Okay, welcome back from lunch. Next up, we have Andrew Hickl. He's the Chief Technology Officer at the Allen Institute, where he leverages AI to solve all kinds of problems, including those involving natural language processing. And so he's going to give us our next keynote.
Excellent. Can you hear me okay? I'll take that as a yes. So, hey, I'm Andy Hickl. I'm the CTO of the Allen Institute. And I'm really honored. It's really an honor and a privilege to be with you today. I am so bummed that I'm not there in Kansas City with you today. I broke my foot in the lamest accident ever over the weekend, just stepping off a curb the wrong way. My wife reminds me that I'm not 25 anymore. And so, unfortunately, the last thing I need to do is travel on a moment's notice with a broken foot. So you'll forgive me for not being there in person. But I have to express, underscore what a special honor it was to receive an invitation to speak to all of you, and really excited for the team to actually help me present virtually. I was honored, but I was also a little bit confused because I am, full disclosure, not a biologist or a curator, but I'm an AI guy. I've been doing it long before it was cool. My PhD was in the late 2000s or early 2000s. But ultimately, the invitation made a lot more sense when I was asked to talk about the impact of NLP on biocuration. And largely because, despite that I now it's safe for me to describe myself as an AI person, I resisted that label for most of my career because I was really staunchly a natural language processing person. And I spent the first 10 years of my career shipping software to government, whether it was question answering or information extraction or tagging or ontology creation work. We had the temerity to replace PCFGs with machine learning and ultimately to make it work. Later on, I had the good fortune to work with Paul Allen in multiple guises and was able to be a part of the team that co-founded the Allen Institute for Artificial Intelligence, or AI2, which we'll talk about a little bit later. And later still, I actually worked at Microsoft, where I was the PM in charge of bringing OpenAI's products to market via Azure. And so ultimately brought them to the public cloud and ultimately to the government cloud as well. And so these days I'm firmly in the biology space because I think it's the best place to be. And I'm leading AI and natural language processing investments here at the Allen Institute. So today I'd love to take my time. Sorry, I need to advance the slides here. I'd like to take my time making, at the risk of being uncontroversial, trying a new framing for my argument today. And I'd like to argue that biocuration is the best example of successful AI transformation in science today. And the people in this room should be applauded and lauded for really kind of fighting the good fight over the last 30 years. Whether it was from quality assurance to HCI to trust and ethics, biocuration should be studied as the canonical example of how to adopt AI in science. And believe me, there's a whole bunch of we don't necessarily know exactly how to adopt AI in science or to really affect AI transformation in science at scale. There's a lot of enthusiasm and a lot of kind of interest in doing it. We'll talk about some of the work that we're doing both here at Allen Institute and at AI2 later on in the talk. But ultimately, we shouldn't have to look any further than the work that's been done by Biocuration to actually talk about how to adopt AI and actually how to do it right. And so my goal today is to really convince you that the last 30 years of biocuration is really one of the best examples. And I may be preaching to the choir, but I'll run the risk to do that. Ultimately, you may ask, why do I care so much about AI transformation? Well, it's my job at the Allen Institute. We have 6 institutes, 6 science units, or 6 institutes that we have across our investment strategy, almost 1,000 employees, and 250 of those identify themselves as engineers or data scientists. And so they're my team. And our real job is to really create the kind of software platforms and infrastructure that really helps them, helps the science get done or ultimately translates data into insight. The second part of my remit in terms of AI transformation is really to kind of create a first-class culture of sense-making at the Allen Institute. We've been known for the last 20 years as a world-class data collection organization. And we really realize that that's not enough to be able to actually be relevant going forward in science. We need to make sure that we're not just creating the world's greatest data sets, we're actually turning them into things that, building the tools that allow you to impute, fill gaps or impute data where it hasn't been collected, really kind of understand the value of different data sets, and ultimately really make sense of it and answer the questions that we all want to know the answers to. And later on, I'll talk a little bit about a new institute that we're creating called Studio D3, which really looks at a new way of doing AI transformation using theory as a part of the virtuous cycle And we'll talk a little bit about how we're trying to get that off the ground. But today's topic, really, we're going to talk about AI transformation and how NLP really affected that in biocuration. And so we'll start by talking a little bit about what is actually exactly AI transformation and how scared we should be of it. My kids are 9 and 13, and they have gone from thinking that I'm a hero because I get to work on science fiction, cool stuff like AI, to ultimately not sure exactly what they think about me because they're afraid that I'm going to be limiting their job options when they graduate. And so, I mean, this is a very real concern even in our household, where we have 2 AI professionals going to be heading the household. We'll talk a little bit about what are the hallmarks of successful AI transformation, and we'll lean on a study that I led at Accenture when I was the chief data scientist at Accenture most recently, that we did in 2023, that interviewed more than 1,000 CXOs about exactly their AI roadmaps and their interest. And basically, we'll take the lessons learned from there. But no surprise, we're going to draw a through line to saying the hallmarks of successful AI transformation that's worked across multiple industries. We looked at more than 25 in that survey. Really are embodied by the work that we've done in biocuration over the last 25 or 30 years. Then we'll take a little bit of a, Billy Joel, we didn't start the fire kind of tour of the history of biocuration and its parallel path with NLP. And then we'll close by talking about some fun stuff. Really what's next and where we should look for continued transformation now that NLP is more or less it's a mature technology, if not solved, as much as it pains me to say that. All right, well, since I'm not with you today, I was going to start by seeing a show of hands, and unfortunately I can't see you, but ultimately, I think there's 2 questions that we wrestle with in terms of AI transformation. Concerns about that AI is going to eliminate your job in the next 5 years, or or more, if you're more optimistic, that AI is going to change your job in the next 5 years. And I think really what I'm going to argue is that, there will be some job elimination, but ultimately most of the work that we're going to be thinking about is really transforming the capabilities that we have. And I'm going to underscore that, really try to drive home the point that transformation is not necessarily a bad thing, and ultimately there's benefits that far outweigh the kind of drawbacks in the short term. So if you're worrying like my kids do, you're not alone. 42% of workers globally believe that AI is going to replace some of their job duties in the next 2 to 3 years. And given that this is old data, some of this has already come home to roost. But the best example that I can give of these kind of concerns that I have that are out there was in one of my senior colleagues at the Allen Institute, a gentleman who runs one of our science units. He came to me and said, "I'm not sure I like you here. My RAs are afraid of AI, and I'm not sure I like it either. Can't we get back to good old-fashioned science?" And I spent a bunch of time kind of talking him into understanding my point of view and ultimately understanding why transformation is not just inevitable, but it's ultimately going to be a beneficial thing as well. We say this, not to buy into the hype, because not every industry is ready for AI transformation. That survey that I talked about, it was really a bimodal or a normal distribution in terms of the way the total readiness of the industries in terms of the decision makers that we surveyed. 23% of the decision makers really called out that or 23% of the jobs that we talked about were at high risk for automation. This is technology, coding, call center work, and 19% was at low risk. And this is more physical plant work, construction work. But the preponderance of evidence was, hey, let's see what happens. We're not really sure exactly what the use cases are, and we're not exactly really sure about what the trajectory is ultimately going to look like. And so 50% of the world is still trying to figure out what their AI transformation path looks like. And science writ large is part of that 50%, with the exception of the kind of patterns and practices you've laid down in biocuration over the last 30 years. Ultimately, I'm not going to belabor this argument here because this is not a conference on AI transformation. But really the value that we see in AI transformation comes from the fact that AI can shift roles and really refocus humans on higher order or more strategic tasks, and ultimately create more value creation in the consulting speak. And one thing that I found that was interesting in some of our research at Accenture, we showed that if you the threshold for actually unlocking well-being and better mental health in workers was really just automating about 30% of the repetitive tasks that they had to do. And really, the outcomes were lasting in terms of job satisfaction, worker health, and ultimately not just physical health, but mental health as well. And so you really, this is going to be the story that we're going to come back to over and over again in terms of the fact that this, we're adding strategic value, we're adding more insights per dollar or per minute in terms of the work we're doing by driving automation using AI or natural language processing. But really, the impact is how do we change the way of working? What is the teaming culture looking like? And really, what is the nature of the kind of the new relationship once AI is established as a partner in the process that we're looking at? So in that survey that we ran, we had about 26 or 30 different hallmarks of successful AI transformation. And I'm going to just focus on 4 or 5 here that we can actually talk about. But the priorities for reasons why decision makers decided to take on AI transformation as a kind of strategic initiative was, they were looking for more efficiency and productivity. They were looking for better interfaces between humans and computers. They were looking for more insight or more value creation. And in scientific context, we can talk about that as when more hypotheses proven or disproven per week or per month or per quarter. They were looking for personalized experiences that better fit their use cases. And believe it or not, they were really actually even looking for employee or worker well-being. And so, what I'm going to argue in the next couple slides, and I won't make too much hay out of this, is the fact that we can make a case that biocuration, as it currently stands in 2025, is really is you leverage NLP to be able to actually deliver a lot of these value, these hallmarks of successful AI transformation. So not to belabor the point, but tools like BioBert or Spacey really are not just driving automation, but they're saving time and effort. And ultimately, and as we think about operationalizing these systems, they're moving more towards real-time analysis and insights and faster decision making. We can talk about cost savings. We can talk about elimination of the mundane tasks. We can talk about kind of changing roles. But really, the value that we see, especially as we think about ways we integrate AI in terms of the work we do in our labs at the Allen Institute, is really how do we shorten the decision time or the time between data collection and decision, and how do we take that down from hours, from weeks to hours to minutes, and really AI is and not just natural language processing, but AI in all of its different forms. It's important for us in terms of how we think about that. This is when the human there's a lot of talk about HCI and what the nature of AI collaboration is going to look like. A lot of the active learning loops that have been kind of so hallmarks of the biocuration work in natural language processing and the kind of co-curation work between machines and humans has really kind of set a standard for the way things should work. But I think the thing that I get excited about, especially as I think about the kind of more sophisticated agentic systems that I've seen deployed in real-world applications, and here I'm thinking about places like the U.S. intelligence community that I've worked with. There's a kind of psychological shift in terms of once AI agents actually have gone past the probationary period and actually shown that they're valuable, that they create an independent value of their own, and once the teams get past the inherent skepticism of technology for technology's sake, but ultimately in teams in the USIC, we find the analysts are actually renaming or giving nicknames to the agents that they use. They're actually embodying them with personalities. They're looking to create avatars and signs for them, faces and decorations for them at their workstation. And in fact, even some of the IC teams that I've looked at, they've actually set aside different parts of their cubicle for them to work in, most likely they're human coworkers. And so really what we're thinking about is in terms of, as we get the collaboration right, we're actually not just thinking about a tool, but we're thinking about a partner that we can actually use in terms of this HCI interface. And then, biocuration has really kind of set the standard for what, with AI collaboration, partly out of necessity because the NLP systems for the last 20 years have not necessarily been perfect or high enough quality or reliable enough to actually allow you to trust them without oversight. And so that quality assurance loop that you put together is really kind of planted the seeds for kind of larger scale HCI kind of relationships and the kind of things that the team relationships that I've just talked about in terms of the IC. I'm cognizant that I spent a year being a professional consultant, and so my speech is forever infected with the value creation terminology. I won't hammer this home, but what we're seeing in terms of successful deployment of AI is really we're shifting the kind of tasks from rote mundane tasks to kind of more strategic critical thinking and high level judgment tasks. And we're even seeing this with data scientists in terms of the work that we do in terms of our labs. We've got a project with AI2 that is actually creates a set of agents to kind of automate traditional data science tasks that are done by data scientists attached to labs. And so instead of actually tasking somebody with a week's worth of work of mucking around in pandas, you could actually task a large language model or an agent-based system to do some of the analysis for them. That doesn't mean that we don't need that data scientist anymore. She can actually then be retasked to think about more critical thinking or hypothesis generation or other kinds of tasks. And biocuration is really kind of, and the work in terms of any kind of information sciences that we've been focusing on have really changed the kind of way that the nature of that thinking or that pattern. I think personalization is pretty self-explanatory. This is something that's important for a variety of industries, especially retail and customer service use cases in terms of AI transformation for other industries. But ultimately, we're really thinking about as agents or more kind of AI-powered systems kind of become de rigueur. We're thinking about people don't just want a one-size-fits-all system. They want the co-pilot or the kind of agent that they interact with to really know what they're dealing with, have some shared history and some sort of personal context. And that's really powered by the knowledge creation, whether it's structured knowledge that you're able to extract from unstructured text, or whether that's kind of contextual knowledge that you're pulling out from somebody's situational awareness. And I'm most fond of the fact that AI is not just going to come for some of your responsibilities, but also it's really helping us reduce burnout and improving job satisfaction. And so, I was at Microsoft when we used natural language processing to kind of employ to change the nature of content moderation and really kind of just reducing the number of kind of potentially questionable kinds of things that people had to look at really was important in boosting job satisfaction and mental health as well. So I think I've made the case probably too long in terms of arguing that the biocuration is a positive example of or successful example of AI transformation and should be something that we mimic across science. And we'll talk a little bit at the end of the talk about how we're actually going to go do that. But ultimately, I was remiss to the invitation that I received not to just talk about how we actually got here. And so, without getting into too much detail, because I could give a 12-week course on how much the kind of parallel paths NLP and biocuration went on over the last 30 years. I'll hit on highlights over basically five distinct phases, and we can argue in the question period about the time, the dates that we associate with each of these phases. But ultimately, in the beginning, there was rule-based systems that really relied on manual patterns and early automation and enabled early automation. They gave way to kind of the first statistical models where we actually used, preponderance of data to actually start informing priors, whether that was, WFST kind of or PCFG kind of grammar-based systems, or it's very basic hidden Markov models, to the kind of first machine learning systems where we had to exploit labeled data and using hand-tooled features. Quickly that gave way to, as essentially that the statistical model period was really valuable enough in terms of actually helping us understand what tasks to be performed, whether it was basic knowledge extraction like named entity recognition, or relation extraction, or event extraction, or things like higher order inferencing like textual inference or question answering or summarization. So ultimately the statistical models, although they never really achieved critical mass in terms of performance, they were really important in establishing the kind of use cases that we know and love today, and that we all use ChatGPT or the like to actually use as part of our either hobbyist use cases or actually putting things in production. Those kind of early statistical models gave way to kind of the first machine learning models, and then quickly, things like MaxN and CRFs and SVMs, and those really quickly gave way to deep learning approaches, especially as compute became available and data became available to actually train the models. And then last but not least, things really changed in the mid-2010s, especially as we were able to promote contextual context-aware understanding using transformer models, which really were empowered by the fact that more and more data became available and tractable, and more compute became elastic enough to actually posit large language models. And so, just to kind of talk about each of these phases in specific, I think that what I'll do, instead of actually going through the history, it'll just kind of draw all the parallels. I think, biocuration, you can argue with me in terms of, lagged 5 or 6 years behind the kind of state of the art in terms of natural language processing in its early days, and very quickly caught up in the 2010s, especially as the kind of dedicated resources and shared tasks actually became to the fore. But in the beginning, there was rule-based systems and manual workflows, and fabulous resources like Uniprot and Firebase, and making sure they have vocabularies and doing the knowledge engineering necessarily needed to be able to kind of automate the information extraction. We talked about this a second ago, but the early attempts at automation really were available unlocked by kind of having critical mass in terms of corpora, unstructured text, whether it was the New York Times corpora or the Brown corpus, and really allowed people to actually spend time hand annotating data sets in natural language processing to make sure that you could actually perform tasks like part of speech tagging or basic syntactic parsing or even semantic parsing in the early days. I think the same thing happened in terms of MetaMap and Textpresso with biocuration, and especially even as we thought about more domain-specific natural language processing models, whether that was for healthcare or EHR, or whether that was for literature review. But ultimately, really, the first kind of systems capitalized on the fact that we were able to kind of have these top-down knowledge engineering systems that were able to project into the text, and then use that to be able to actually extrapolate the kind of priors that we learned from the annotated text we had to the unannotated text, and really create a virtuous cycle loop of knowledge creation over time, especially tagging. This is the era that I lived, most specifically in terms of the research that I drove, really was around information extraction and then textual inference. And I can't underscore the importance, not just of the machine learning, basic machine learning, whether it was MaxN or SVMs or CRFs. It was lots of different ingredients you could throw into the soup. But really the kind of thing that drove and catalyzed a lot of innovation was shared tasks. And biocreative was really an example of that. The MUC, MET, CODL, TREC kind of evaluations in domain general, and to some extent some domain-specific information extraction or contextual reasoning tasks help drive this. But ultimately, having a benchmark and in some cases having summertime evaluations, shared tasks that everyone can lock their system and run it on unseen data, really catalyzed so much innovation and really helps kind of drive science forward in interesting ways. But ultimately, that really started and ended with the information sciences work that we had to do in terms of creating knowledge resources up front, whether it was creating corpora or ontologies, making sure that those actually got mapped down and translated into resources that could be made computable. To go more quickly here, the first part of the last decade, we were still very much in the deep learning world, and just getting over the fact of being liberated from the fact that we had to do manual feature development engineering. And for the well-heeled customers amongst us, or the well-heeled researchers amongst us, we were able to actually start playing around with neural models and really kind of getting excited over the fact that it wasn't just more data that was driving performance increases, but actually new model architectures drive that. And I think this is where biocuration really caught up, and really was the fact that you were able to create this virtuous cycle and have curators in the loop and create knowledge really allowed you to kind of make sure that you were able to take advantage of not just deep learning systems or traditional machine learning systems as well, and really kind of accelerate the amount of knowledge creation and tag corpora that you could actually use. And then, everything changed in the late 2010s, especially as we think about, kind of attention-based infrastructures and transformer models became more and more dominant. And then it really kind of raised the question of, really, could you rely on one model to rule them all, or did you actually have to do domain customization in the way that you would have to do in the old days? And ultimately, the jury is still out in terms of how much domain customization or fine tuning needs to be done, especially as you think about high quality or high kind of reliability tasks. Always some fine tuning is always valuable in some cases. And the question for us is really now is especially as we kind of think about operationalizing some of these more lightweight models, because we don't have necessarily always rely on the super large language models. Real challenge ultimately will be, how do we operationalize these kind of these transformer? How do we operationalize the increasingly large domain general models actually perform more domain specific tasks? And again, here, kudos to the biocuration community for being ahead, not just in terms of the tasks that you were able to extract, whether it was relations or events, things that really I fought for over the years in natural language processing, but never really took off. But ultimately, in terms of the kind of real insistence on having domain-specific models that just had to work in lots of different cases. Just to kind of put a capstone on it, we're very much in the age of kind of super large language models that purport to do everything for everything for everybody and kind of support every possible use case. I think we're seeing some catching up in terms of the kind of large language models or the kind of foundational models for other biological tasks. There's a real kind of debate that we have internally in terms of ChatGPT was able to kind of consume all of human language and perform a bunch of tasks without any kind of use case specific training or any use case specific development. Can't we do the same thing for biology and really create foundational models? We just dump all the omics data we have into one big model and let the model-based architecture, the foundational model architecture, sort it out. I think the cooler heads amongst us really believe in the fact that use case driven development is really still going to be important. And ultimately, we're seeing that in terms of the path to making sure understanding how much use case driven development is important really kind of runs through validation and understanding where the performance of your models at a very detailed level, not just in an extrinsic analysis in terms of an end-to-end performance number, but an intrinsic analysis in performance of evaluating the performance of the model or the AI system on every specific task in your pipeline. And again, kudos to biocuration, and one of the kind of things that we should be thinking about as we kind of mimic the biocuration workflows or the history in your path through AI transformation into science all up. We should start to be thinking about what role validation and triage actually plays in terms of not just in terms of performance evaluation and vetting something for production, but also understanding whether there's some interesting data curation or kind of fine-tuning kind of pre-preparation that needs to be done for fine-tuning as well. So anybody who tells you they know what's next is lying to you or is trying to sell you their startup. So I'm going to talk a little bit about some of the things that I get, the most popular ideas that people kind of pitch to me. But what I'm really going to quickly pivot to is some of the work that we're doing together with AI2, our sister institution, in terms of actually trying to kind of figure out exactly what the next phase of AI transformation ultimately looks like. I really believe that the kind of drive in terms of curation, especially given some of the things I see, especially in other domains like financial analysis and oil and gas analysis and other places where knowledge extraction from unstructured or structured data or a combination of both is important, really is driving towards autonomy and really having agent-based infrastructure that can actually perform a set of highly defined and well-defined tasks reliably. And then whether that's reading or data transformation or ETL or basic data analysis or data frame construction. And then so we're seeing workbenches and tool sets of agents kind of established today. And really what we're looking for is the next step is trying to figure out how we can make those sets of agents operate in consort and kind of anticipate the kind of actions that you're going to actually perform on their own. And so the question for us is how much human oversight is needed? And my science leader that I talked about before would insist that, that there is no task that could ever be performed without human oversight, even in basically ETL kind of use cases. And I've got other leaders in my community who believe that they're okay with certain kind of given certain assurances, they're okay with more and more automation. I think one of the things we're really excited about, and I had a good fortune to hang out with Peter Lee, who's the president of Microsoft Research here at the Allen Institute not too long ago, and he joked that if he sees one more lab in the loop presentation where the AI is going to powering a virtuous cycle, he was going to throw up on the next person who presented it to him. But really, I think there's some value in terms of thinking about experimental design assistance and really kind of mining literature for hypotheses, kind of figuring out exactly how to kind of generate protocols from those hypotheses, and then designing experiments, and then ultimately with appropriate APIs, the Hamilton equipment, or your equivalent, your favorite kind of lab automation hardware, could you actually start automating some of those experiments and really kind of reducing the cognitive workload that an RA or somebody in the lab has to go through. I'll close by talking about a little bit about some concrete things that we're doing with AI too. I think the jury is still out in terms of, what does it mean for AI transformation for science all up? And we've talked about some of the concerns. We've talked about some of the kind of hallmarks of what it would take. We talked about some of the positive things that the biocuration community has done over the years. And this is an example of Peter's dreaded lab in the loop slide. Really what we're thinking about is less about the kind of operationalized. I think that each of these investments, whether it's this retrieval and synthesis of literature, hypothesis generation, or planning experiments, or even generating papers, I think that there's reason to believe that there's going to be some automation done for each of these kind of steps. The real challenge ultimately for us is going to be establishing how much automation, how much transformation is going to be needed, and how quickly do we go. So part of the reason we work together with AI too is we really want to make sure that we're not just developing AI in isolation and throwing it over the fence and hopefully seeing what was valuable for the lab and then complaining and basically throwing back feedback to the team. We're actually doing this in concert with with AI developers as well. And so that's part of the reason we stood up our own AI community here at the Allen Institute. And AI2 standing up their own biology experts as their team, so we can actually cross-pollinate ideas and work on use cases together. And it's not just AI that's kind of driving the development of our possible use cases. I talked a little bit earlier in the beginning of my talk about Studio D3, which is one of our new institutes that we're getting off the ground this summer. Really looking at how can theory be a way to constrain the hypotheses that we consider when we generate an AI. Sorry, when we have an AI system that's able to generate hypotheses, how can theory constrain the kind of sets of outputs that the model can produce? And then ultimately, how can theory be used to actually help interpret the predictions that come out of a model that's driving analysis? And so you really think there's really value in terms of, as AI becomes more important, we need more and more powerful. We need to make sure that we have not just generic human oversight, but we have principled human oversight in terms of the way we actually think about governing this work. But also, really, the flagship platform that we're building together with AI2 is called Nora. It's otherwise known as the Never-Ending Open Research Assistant. And the dream here is that we have a set of agents. We embody this with one assistant with a mortarboard right here in the middle, who's able to communicate with so that was the person working in the lab, understand contextual awareness of what they're trying to do, and to be able to actually draw structured, draw knowledge resources from a variety of sources, whether it's code, whether it's workbenches of agents, whether it's literature, or whether it's just the generic web. And so again, we have to be use case driven in terms of our approach, especially to be able to learn over time. But we've got, if anybody's interested, you can contact me after the talk, and I'll make sure that I can actually add you to the beta for Nora. We also have use cases like summarization, where you can actually understand your contextual query. Sorry, not just have to specify, hey, summarize this paper, but we can actually understand what you've been working on, what kinds of questions you were worrying about, what kinds of things you already know, what kinds of things you don't already know, be able to kind of generate summaries that are kind of cognizant of what your information gap is, and be able to provide the kind of gap, the actual information that you're looking for or that you would need most critically, as opposed to just scattershot giving you all the information that's in the document. Literature comparison, I think, is really interesting kind of approach. Be able to actually take results, especially build your own kinds of survey papers from the sets of literature that's out there today. That's something that's relatively mature right now. Question answering, which is where I made my bones early on in my career. I'm still fascinated by the fact that we can ask not just questions like who is Tom Cruise married to, but we can actually ask questions about how would I design a better protocol to be able to achieve something based on these 5 different papers, and be able to get a structured answer that makes more or less more sense. And again, validation is going to be an important question, especially as we think about coming up the stack in terms of identifying harder and harder tasks to perform. I've alluded to this a couple times in terms of one of the way we challenge our work and we do evaluation for the work we do, as we say, can you actually replicate the analysis that was done by a paper? If all you were given was the actual paper and the data set that the paper was actually generated from as your set of instructions, and can you actually generate not just the analyses in terms of textual hypotheses or textual conclusions, can you actually generate the figures that people have done? And this isn't just image matching, this is actually inference-based kind of analysis. And so there's a lot of excitement here. Ultimately the use case, the dirty secret here is that it only performs about 40 or 50% of the time. And it really works well with cherry-picked examples. And it's ultimately only as good as the quality of the paper is that was written. So we're far from actually being able to have really unlimited or unsupervised kind of knowledge acquisition systems that are able to kind of produce large-scale kind of knowledge. But we're taking important steps towards the next direction. I'll skip this in the interest of time, but I think ICLR this year had 3 papers that were accepted that were written by agents. We're going to get to the more. This is not just a question of when or if. This is a question of when, in terms of where we're going to be in terms of creating papers from data. And again, I think the jury is really still out in terms of validating, what and kind of making sure that these things are hallucination free, and that the chain of reasoning that they actually produce to kind of support their answers is really valuable. The last couple of minutes that I'll talk about here, we've got, there's many open questions to still figure out. I think, I haven't spent enough time. I've talked a lot about validation. I haven't spent enough time thinking about trust. I think trust in science is at a critical moment all up. We need to make sure that we're being very careful in terms of understanding how we think about deploying these systems in a way that would improve reproducibility of our science, and not open the door to detractors, making sure that are arguing that our work is even more opaque than it is perceived to be. I think the human interaction piece is interesting. We talked about the teams that the intelligence community created around AI. I think a lot of the interesting challenges there for us would be, what is the nature of an AI-powered RA or an AI-powered lab? And really, what tasks are we willing to kind of automate? What kind of tasks, what kinds of interfaces are we willing to kind of put up with? And what principles would guide those designs? And then, I won't talk about value, but the last thing is, we spend a lot of time in our community thinking about ethical and responsible integration. Making sure that when we think about when we try to constrain the set of proposals that an AI agent can make, how do we think about making sure that the science is done ethically the first time? All right. Well, I am thoroughly enjoyed the fact that I was able to spend some time with you all today. I regret the fact that I wasn't in person, and I hope to meet many of you in the future. But I think that the parting thoughts that I'll leave you with, I don't think they graduate to the point of conclusions. But ultimately, biocuration should be, the biocuration community should really pat themselves on the back, especially because what they're doing, what you have done is demonstrated, one path towards successful AI transformation in science. And I think really the takeaways here are the fact that the introduction of AI never really didn't just automate and replace human roles. It fundamentally reshaped them. And it was able to do that because you were able to maintain validation and KPIs that steadily shifted from just marking up X number of bytes of text towards actually thinking about quality, judgment, and even kind of new, not just bytes of knowledge or bits of knowledge being able to create it, but also the kinds of insight or categories of insight you're able to produce. And I think, I think really the thing that really helped you be successful in the best case was the fact that, you never were willing to remove humans from the loop, and every chance you got, you were able to kind of further double down on the fact on ways to integrate them more closely into the process, whether that was in terms of creating the ontology up front, or creating the knowledge sets, the data sets from that knowledge, or whether it was driving validation at scale, or whether it was actually trying to drive continuous learning through active learning, or human in the loop kind of processes. And so really, I think there's a lot of excitement to think about. Really, congratulations to be received for the work that you've done so far. I think the interesting thing for us as a community is going to really be thinking about what the next 10 years hold, especially as the reliability of systems get more and more powerful, and the human relationship with the AI is going to become even more critical to get right. So thank you so much for your time and attention. I really appreciate it, and I think I've got time for questions as well.
Thank you very much, Andy. We have time for some questions. Is everybody AI'd out already?
So you mentioned that AI will help people shift their work from more grunt things to more valuable things. To what extent do you think that'll affect everybody? And you mentioned it will increase quality of life by sort of freeing you up to be more creative. But as you become more creative and more valuable, do the expectations scale so that you're still sort of miserable because you're under an increased set of expectations?
I'd like to think not, but it's definitely a risk. I think ultimately the narratives that we get most excited about in terms of AI transformation are ones where we're empowering people to do more. And but ultimately at some point there's a cap in terms of some of the tasks that need to be performed. And I think you have examples like the call center work. It was really kind of early adopters of a lot of the AI transformation that we're seeing in terms of driving automation, and and the parallels between the call center work and some of the curation work is interesting in terms of hey, what was really powerful in kind of enabling the call center transformation was large-scale knowledge extraction, contextual awareness, making sure the humans could actually validate, stop from kind of creating knowledge to actually validating the knowledge that the system produced. And so, but also what we found as more and more call centers became automated, there was a cap in terms of the task. At some point, the job still was somebody managing, customer service calls. And so I think one of the things that I get excited about biocuration is the fact that, the sky's the limit in terms of the way we can think about it's truly an analytic task. And so, how do you think about kind of expanding or extending the knowledge that somebody's able to produce from something that's assisted in ontology or a fact, or kind of validating a finding, to actually thinking about actually being part of the creation process of the protocol to design and capture that finding in the first place.
This was very interesting. Thank you so much.
Thank you.
This was also very timely for me because just at AAAI, we talked a little bit about, the applications that industry is building, and also open source versions are in progress too for, helping scientists do science. But as we went into discussion sessions, one of the things that was really hard for me, it was my first time thinking about it, was to see where the line is between me being a scientist and outsourcing something. So I'm wondering if you had any thoughts on that, and where that line may be, in addition to all the ethical, questions and the concerns that come with it.
Yeah, I mean, I don't think anybody has a good answer for it. I don't think there's a one size fits all or kind of universal answer. I think, one of the things we've been wrestling with is really just kind of understanding use cases. I'll give you an example. We turned on Copilot for all the institute here, Microsoft Copilot for all the institute here, and people immediately started asking, their supervisors, was it okay to use Copilot to write an abstract for their paper? not writing the entire paper, but ultimately just write the abstract for the paper. Who hasn't spent that slapdash, put a slapdash abstract together at the last minute, and you really that put together really kind of opened up a can of worms in terms of how we thought about, where did that line actually fall, and ultimately some people were saying, hey, it would be okay for the co-pilot to write the abstract with the appropriate attribution. Some people said it was not appropriate at all. Some people said it was just fine. And so one of the things we had to really figure out was, what do we, when we stand behind our science, or we stand behind the kind of quality of the ideas that we're putting together, how much augmentation are we willing to kind of put up with? And I think really, I mean, there's those set of questions that are out there. There's other set of questions in terms of how valid is the use case to be able to actually support the science, AI integration use case in the science. And so really the second one is, I think the first one, we're going to come to some principles around what's acceptable and what's not acceptable. I think the second one's going to be interesting in terms of, because it's going to require us to have really strong validation criteria, or better understanding of exactly what the impact would be. And that's going to have to come down to KPIs and real metrics in a real way.
Okay, we have time for one more. Otherwise, we'd give a big thanks to Andy.
Thank you, everybody.
So this is the beginning of our natural language processing session, and the first speaker in the regular talks has been Gyori.
Okay, so I will talk about a project that's actually a collaboration between my lab at Northeastern University and Livia Perfetto group in Sapienza, who are building the senior pathway database. So the big picture here is that analyzing large data sets can really benefit from structured prior knowledge that characterizes the complex behavior of biological systems. So the question is, how can we scale up structured mechanistic knowledge to a point where it's effectively able to be applied to large-scale omics data sets? Pathway databases that are curated typically from literature by experts are an important component of this. Senior is one of the examples of actively curated pathway databases, where this is a page for a protein called PPM1D, and you can see various relationships, interactions and regulations that have been curated for it with appropriate evidence linked back to literature and specific sentences. But given the scale and scope of literature and the pace at which it's produced, 4,000 to 5,000 papers per day, the question is not only how you can actually monitor that literature and keep your pathway database up to date, but also how you can, in a systematic way, tell what is missing from your structured resource, like what might still be out there that hasn't been curated. So that's a difficult question to answer. And so my lab has been developing a knowledge assembly approach that relies on the processing and alignment of both structured and unstructured sources, structured sources being pathway databases, unstructured being primarily literature, with multiple different NLP approaches to extract what we usually call causal mechanistic statements from literature that then go into a layer of knowledge assembly. There's an internal knowledge representation, which we call Indra statements. And then on these Indra statements, we can perform a number of different knowledge assembly tasks that have to do with correcting certain systematic errors, finding and resolving redundancies in a principled way, assessing believability or confidence, and in some cases, inferring missing information based on pieces that are there. And this pipeline, normally, it extends further than just knowledge extraction and assembly into modeling and data analysis. But for the purposes of this talk, we are primarily using the first part of this pipeline, in particular, the literature assembly piece. So you can imagine how all the different structured sources, as well as different natural language processing systems, they produce or they represent mechanisms in very different ways, in different formats. And so one of the tasks that Indra does is it normalizes these variety of representations into this Indra statement representation. And you can see one example of an INDRA statement that's a phosphorylation statement here with an enzyme and the substrate both being grounded entities. So they have database identifiers with some mechanistic detail like a residue and a site. And there's also an evidence data structure that keeps track of the full provenance of where everything came from. One particular issue that is important for this project that INDRA addresses is that there's actually a widespread issue with references to amino acid sites, both in literature and structured databases. So pathway databases also have this problem. For instance, there are two canonical phosphorylation sites on the protein ERK, and it turns out that there are more references to incorrect, that is not reference aligned sites for these phosphorylation sites on ERK than the real ones. And so we built out this machinery to account for different types of systematic errors that cause misalignment of phosphosite references. These include, for instance, the cleavage of the initial methionine that has an off-by-1 effect on the site position, cleavage of signal peptide for receptors, accidentally referring to mouse sequences instead of human, and so on. There are multiple different variants of this, and there's this pipeline that accounts for these various possible issues and normalizes sites. And there's actually a dedicated package for this called ProtMapper. It's integrated into INDRA, so we can apply it at the statement level to fix or normalize phosphorylation site positions and other PTM site positions that appear in INDRA statements. And so at the end of the day, I won't go into all the methodology related to this, but we get these assembled statements where there's a single representation of something like a ubiquitination process with all the corresponding evidence, both from text mining and from pathway databases aligned in a kind of single package. We've scaled this up to basically encompass all available literature, abstracts, where that is available, full text where that's available, and combine that with the content of various pathway databases and build a queryable knowledge base. This is a fairly complicated and actually expensive infrastructure that's set up in the cloud on AWS, and it can self-update autonomously, so it reads new literature every day and adds that to the database, and you can go in and query it at db.indra.bio. There's also a very good REST API, so you can query it programmatically. And so in a paper describing INDRA a couple of years ago, we looked at two main use cases. One of them is explaining omics data using automatically assembled networks from literature and databases. But there's also a second use case that we looked at the time, which is showing a proof of principle of how you could use this literature assembled knowledge to extend a curated knowledge base. And in that case, we looked at BioGrid specifically. So we looked at what are the highest confidence statements that come out of literature-derived knowledge assembly that aren't yet in BioGrid. And looking at if you were to curate starting from the top, so the highest confidence statements, what would be your curation yield in terms of correct statements that you look at and curate and can add to the database. And it shows that by going down this ranking of protein-protein interaction with the highest number of evidences from literature that are typically fairly confident, you can get a very high curation yield. And so this leads to an idea related to curation that is mechanism-centric, which is a little bit different from the more classic and more typical paper-centric curation approach. The paper-centric curation approach being you first identify a paper out of the many possible papers that is relevant in some way to your database, and then you curate everything that is in that paper and add it to your database. The mechanism-centric approach, which our pipeline enables, is one where you look at a specific mechanism that hasn't yet been curated. It might have evidence from multiple different sources, multiple different papers, and then you look at the evidence corresponding to this mechanism. Like this is just an example of a phosphorylation relationship, and determine whether the evidence correctly supports this statement according to the criteria in your database. Like perhaps you need a specific primary experimental evidence and so on that you can confirm by going back to the publication, and then that goes into your database. And so we actually employed this mechanism-centric curation approach in this project. And the idea was that we first focused on trying to extend the senior database with dephosphorylation statements, because we found that these are not very well curated. There aren't many active resources that are curating these, and so it's a good first target to pilot this idea on. And this central part is actually important in that we specifically looked at not just dephosphorylations, but cases where in the same paper, we also have evidence for the polarity of regulation that this dephosphorylation induces. So inactivating dephosphorylation or activating dephosphorylation, which is much more valuable than the dephosphorylation alone. And then we filtered whatever we got from Indra against what was already in Senior and created a ranked list of most confident to least confident interactions. And you can see how, as I said, because of this central idea here, when you have a pair of a phosphatase and its substrate, you have evidences both for some kind of regulation and also PTM here, so inhibition and dephosphorylation in this case. We've also built a curation interface that lets you load a custom set of statements, in this case, the set of statements we got for Senior, and curate it through a local web interface. I won't go over all of the details of this, but there are different ways of browsing and searching into these statements, filtering ones that have already been curated. All the entities are grounded, so you can link out to HGNC, for instance, to confirm that the named entity normalization worked correctly. And then you can click on one of the headings to see the underlying evidence and click on this little pencil to open up a little curation form where you can select whether this sentence correctly supports the statement or if there's some kind of an error type. You can also add a comment. And submit that, and then that goes into the Indra database. And once you're done with curation, we can query out all of these curations from the database. There's actually, beyond the scope of this project with Senior, there's actually 16,000 of these curations already by 40 curators. This is actually a relatively small subset of that, so about 700 curations specifically for this dephosphorylation project. Each curation looks something like this, so you can have you can identify who did the curation when, identify the statement that was curated, any comments, and so on. And then we built a pipeline that takes these exported curations and converts it into a form that's directly ingestible by Senior. And at the end of the day, we got 233 new unique TTM regulation pairs that could then be ingested directly into Senior. And this is one example where you see the one line from this spreadsheet that is now on the website as a statement that surfaced with some underlying evidence sentences. So going forward, we've generalized this idea to other PTMs. This is Indra's overall representation structure of different types of statements. There are many different types of PTMs that are surfaced, and we've actually already finished the curation of ubiquitination statements, and we're looking at phosphorylation next. And more broadly, and actually another activity is trying to assess whether this, or to what extent this curation mode is more efficient than the classic approach to curation taken on by this database. And so another feature that I wanted to highlight, and this is actually something that Charlie prototyped when he was in my lab, is we have our knowledge graph extension of Indra that runs at discovery.indra.bio, where you can actually go in and browse different groups of proteins. And specifically, there's a flag here that you can exclude database evidence that lets you actually look at, for instance, kinase substrate pairs that haven't yet been incorporated into any curated pathway database, and it's ranked by evidence. So you see the top one here has hundreds of sentences supporting it, but it hasn't yet been curated. Same principle, you could go in and look at kinases, deubiquitinic enzymes, intrinsically disordered proteins, and so on, and start looking at what are the top candidates for curation. And with that, I'd like to thank members, past and current members of my lab who've contributed to these technologies in various ways, as well as Livia and Marta from the Senior team who did the curation and for various forms of funding that supported this work. I'm happy to take questions.
time for 2 questions. All right, I'll ask one. What other databases in this room do you think that Indra statements could support a similar curation workflow in the same way you did for Synjra?
Yeah, very good point. I should have probably said this explicitly, that I think conceptually this kind of approach could be applied to many other databases that curate similar types of statements. I imagine, BioGrid, Reactome, Wikipathways, and other similar resources that rely on similar types of statements could benefit from something like this. Gene ontology, potentially GoChems, right? So, and we're very happy to partner with everyone and explore these possibilities.
Appreciate the broad range of PTMs that you're targeting. Is there a way to sort of visualize or draw knowledge about how one PTM might affect another PTM, the crosstalk between PTMs?
Like phosphorylation and ubiquitination, for instance, or something like that. Yeah, I mean, so in principle, the Indra statement representation allows you to encode agent conditions that are preconditions of some process happening. So for instance, MAC has to be phosphorylated on a certain site for it to then phosphorylate. But very often these are not made explicit in literature, right? So these complex molecular preconditions of certain things happening are not very well reported and therefore are fairly challenging. But it would be possible to curate these in this representation format, or in some cases to infer these. We haven't looked into the cross PTM issue yet. That could be interesting.
All right, thank you very much, Ben.
Thank you.
Next up is Daniela Raciti.
Thank you, Charlie. Can everyone hear me okay? Okay. Hi, everyone. Good afternoon. I would like to thank the organizers for the opportunity to present the latest knowledge work today. My name is Daniela Raciti. I am a curator at Wormbase, which is a part of and the founding member of the Alliance of Genome Resources. So today I'm going to talk about our latest work, which is about characterization and automated classification of sentences in the biomedical literature. So the challenge we're facing is how do we scale high quality biocuration when the resources are actually shrinking? And people have adopted 2 main strategies. One is community curation. So many groups are asking their authors to flag entities from papers, flag data types, and do some manual fact extraction. And the other approach is artificial intelligence. So our AI is used for, our NLP is used for entity recognition, document classification, and also do some limited fact extraction. So what we're doing at Wormbase, and specifically with the Knowledge Project, we are combining artificial intelligence and community curation. So the acronym stands for author curation to knowledge bases. And this is the, oh, I'm sorry. I pressed the wrong button. This is the original paper. We basically, what we're doing is we're emailing authors shortly after they have published their paper, and we ask them to confirm entities and confirm data types flagged in their paper, validating our machine learning methods. This is to give you an example. So, for example, we extract entities from the paper for experimentally studied, and in this case, experimentally studied genes. And the authors can either remove the entities or add using autocompletes. Another example is data type flagging. So we use neural network to flag the paper for specific data type. We present the form and they just need to validate the results. And most importantly, we have a professional biocurator dashboard where Kimberly Van Ocken and I are validating the results that are submitted by the authors. So now we want to go a step further, and we want to aid authors in fact extraction. And the two use cases that we started with are two relevant data types curated at Wormbase, specifically gene expression and protein kinase activity. So in order to achieve our goal for aiding authors in fact extraction, we first need to optimize training data. And we know that as curator, we know that sentences describing the relevant facts, they can be scattered across the paper. They could be in the results section, in the figure legend, or even in supplementary material sections. So what Kimberly Van Ocken and I did was to create a training data set by collecting all the sentences that a curator might use for curation. We assign sentences to subclasses, and then we confirm the classes via curator cross-checking. So we identified many different classes. We've seen that authors were reporting sentences that directly reported experimental results. And these sentences were either fully curatable, and this is an oversimplification, but it's just to give an example. So in this case, the sentence contained all the information necessary for curation. So for example, SNA3 GFP was expressed in the germline. I'm going to show only for the first example the protein kinase activity because I will not have time, but it will be like this enzyme phosphorylate this substrate. So you have all the information a curator needs. Then we've seen that there were partially curatable sentences. So they would say, the author would say, the translational fusion was expressed in the germline. So you don't know what is actually the protein that was expressed. We've seen that there were summary statements. So these were like either, again, fully curatable. And we found those either in the conclusion or in the abstract or in the introduction. But again, these sentences contain curatable information. It's like in summary, we've seen that SNA3 is expressed in the germline. And we've seen that some, again, were summary statements partially curatable. Our work shows the localization of SNA3, but you don't know where. You don't know where. Summary statements also included previously published results. So like Smith and al. Have shown that SNA3 localizes in the germline, but we may not care about that because it could have been curated already. In addition, and we're getting to the end, there were many classes, but there were other related, language-related sentences. For example, a sentence that contained an hypothesis or a rationale. So we hypothesized that SNA3 was expressed in the germline, but you don't know if then it was actually the case. We've seen sentences that described an experimental setup. It's like we prepared the SNA3 GFP translational fusion by adding the promoter and fusing to the GFP. And lastly, we also have sentences that contained related language, but didn't have any curatable data. For example, you use this SNA3 promoter to drive expression in the germline, but you're using this as a reagent, really. So after this analysis, which was very intense, but fun and interesting, we have selected 589 sentences for gene expression, 494 sentences for protein kinase activity. And this is what we call our gold standard set. So then we thought, okay, what can we do with all these sentences that we have collected and all these classes? And it became clear that we could train classifiers for having for extracting fully curatable statements from the literature, to extract partially curatable statements from the literature, and also to extract language-related sentences. Our goal would be like then to present to the authors or to professional biocurators the sentences that contain fully curatable and partially curatable information. So the overall data set contained the gold standard set that I talked about. We also took 1,000 randomly selected sentences from papers, and Kimberly and I validated those as being positive or negative, and 500 negative sentences. With this training set, Valerio Naboldi, who is the lead developer of the Knowledge project, trained, first of all, traditional machine learning model, such as support vector machine, multi-layer perceptron, logistic regression, and others. And then he determined the best performing one based on the F1 score. And in parallel, Valerio, Cristabone from Frybase, and Michael Muller from Wormbase also fine-tuned large language models, such as BioBERT and GPT-4o, to see how they were performing. I'm putting here the QR code for the GitHub, but we also have a poster, so you can access this data there. So the results of this, we've seen that traditional machine learning models have a large confidence intervals, suggesting that the data is not large enough for the task. While we see that that BioBERT in orange and GPT models, they perform similarly with quite high F1 scores. So we were very encouraged, but one thing that we were worried about is like, now if we're going to try to do this for other data types, we have to ask curator to collect 500 sentences, then they're not going to like it. So we've done a degradation analysis to see how small could the training set be. So we wanted to see if we were decreasing the number of sentences, how the F score and the performance would change. And basically, we've seen that even with 40% of training data, which is about 200, 250 sentences, we had an F score greater than 0.8. Okay, so this is not yet in production, meaning that we're not yet presenting the results to the authors, but we're close. I can access, as every other curator can access this information already in the Knowledge dashboard. And as a matter of fact, I use it during my daily curation tasks. Our future work is to develop a UI in Knowledge to present the results to the authors so that they can easily click a yes and no for the sentences. We want to highlight sentences in the context of the full text because, for example, those experimental results could also be, so like how the translational fusion was constructed. This is some information that me as a curator, I collect. So we would want to have easy access to that information as well. We want to pre-populate curation forms with Go terms, curatable entities. And obviously, we want to expand to other data types and generalize to other species literatures. As I mentioned, we have, I don't know if I mentioned it, but we have submitted this to database. This is accessible on bioRxiv. All the training data is there, so you are welcome to ask questions and use it. With that, I would really like to thank Valerio Naboldi and Kimberly Van Ocken, which are a part of the Knowledge project, and they are sitting in the audience. It's really great fun to work with them. Paul Sternberg, who is our PI, and a special thank you to Cristabone and Hans Michael Muller for the BioBERT and GPT-4o fine-tuning. And thank you very much for your attention.
We got time for some questions.
Working this time? Okay, good, thanks. So thanks again. You know, I was here on Saturday, and I think I'm finally starting to understand this. So this is fantastic. I wonder, I have this idea that the fully informative sentences are perhaps more helpful to you than the partially informative sentences. So first of all, is that accurate?
No, because for example, if you say SNA3 is expressed in the germline, period. And then you say, but it's also expressed in the pharynx, but it's also expressed in the, and is seen in the cytoplasm, right? So that's a partial sentence, but it's contain really key information. So we've noticed, initially we thought, okay, maybe we just have to take neighboring sentences, right? But not always, because you get a lot of noise if you take neighboring sentences. It could be that the previous sentence doesn't have anything to do. We don't want to send too much information to the authors to validate, nor to the curator. So it's like, if you know that this contains partial data, then it's like, you know, and is expressed in the pharynx, then the author would say, oh, yeah. Still, you know, we'll need to pre-populate forms and we'll have to see how that works if the authors are describing expression of 4 different genes and there are some partials, like what belongs to what. You still have to, but it's a start.
Okay, thanks. And you anticipated the rest of my questions, so thank you.
Okay.
I have a question. Thank you. This was really interesting to see. I was just curious about the author's participation in this and whether you've gotten some feedback on like how responsive authors are, how involved. Do you have any advice? Because we have
Yes.
a similar, situation, I guess, where we're trying to always get authors to be involved in this like later curation process, but it can be challenging.
Yes, yes. So Kimberly Van Ocken and I had a whole session, a workshop on community curation on Saturday. All the presentations are available, but specifically for the Knowledge project, the latest statistics is that we have 43% response rate, which is quite high. We started in 2019 from 19%, so we went up. There are some things that we hypothesize that have been working, such as emailing all the authors. We started like emailing only the corresponding author. Now we are emailing all the authors. We go to international meetings and really connect with the community. Again, Kimberly was showing this in one of the workshops, like giving them some prize, giving them the leaderboards. We are planning to collaborate with Apicuron, or we not planning, we already started discussion, like to have badges and medals, so that really like this friendly competition. And we gave a worm trophy, which was very nerdy, but people loved it. So yeah, it's a multi operation, like to engage the authors. But we haven't yet shown them the sentences. We hope that this helps, because now we have a free text. So the neural network will say this paper contains gene expression data. If it's pre-checked, they just don't do anything. But if they want, they can submit a sentence or the paragraph. And I do receive it. So they take the time to copy paste. If we present it already and they don't have to do, hopefully they like it better. Thank you.
All right, thank you, Daniela. Next up is Anaïs Mottaz.
So hello, everybody. I'm Anaïs Mottaz from the Swiss Institute of Bioinformatics. And I'm going to present the work of my teammates who could not attend the conference. So I do my best to present their work clearly and to answer questions. So I'm going to present their efforts to enhance the SIB Literature Services to support biocuration. So first, what are the SIB Literature Services, or SIBLs? It's like an umbrella over a set of text mining services that we have developed. And that includes search engine and triage engine of scientific literature in the biomedical and biodiversity domains. I can cite, for instance, Biotic Explorer for searching biotic interactions in the literature, or Variome for searching variant and disease in the publication. I had a poster yesterday. We also developed Cell Triage. It's a triage engine based on LLM to retrieve publications that are relevant for curation the cell lines. So that was developed for Celosaurus. And finally, we have Biodiversity PMC, which is the core of SIBLs. So it's the core of SIBLs because it's where all the other services retrieve the data and the annotations. It offers a set of many documents, including all the abstract from Medline, all the full text from PubMed Central, all the supplementary data that are parsed also to be searchable, also other biodiversity journals that are not in PMC, and finally the taxonomic treatments from PLADI. For all these documents, we offer search methods going from simple keyword search to question answering. And finally, all the documents are semantically enriched through automatic annotations to existing terminologies. And this is what I will talk about today. But just before presenting the terminologies we use and the workflow and the evaluation, I just want to give an example to just to remind why mapping to terminologies are useful. For instance, when searching for, I don't know, a taxon, they're linked to terminologies and able to find not only publications that report the taxon, but also all the species belonging to the family, in this case, Theropodidae, like flying, and all the synonyms that can be used, like flying foxes or old world fruit bats. So we cover many domains with the terminologies, like molecular and cellular biology with the cell lines, gene ontology, gene and protein names, also disease, drugs, and also in the environmental biodiversity domain, we also have plant ontologies, taxonomies or relation ontology that we use in BioExplorer. So the workflow is the following. Every day, all the new documents from Medline, from PubMed Central, from Plazi, and including the supplementary data, are retrieved, parsed to be uniformized, and then annotated, and then are directly available through our APIs for searching the documents or fetching the documents and all the annotations. And we also have web portals like the general biodiversity, the main biodiversity portal, and also the BioExplorer Varium ones. So all these documents represented in December 2024, because of course it increases every day, 73 millions of documents and 15 billions of annotations. So as I mentioned, all the documents, you can search the documents, but you can also fetch them and retrieve all, for instance, all the publication or subset of publications with the annotations. And it's available in the BioC format. And it's hierarchically organized, split into sections, paragraph, and even sentence level. And the annotation are done at the sentence level. So for each annotation, you have its position in the sentence. It's the linked concept, the concept term, and of course the terminology from which the concept comes from and version. So we can make the annotation traceable. Here is an overview of the percentage of annotation per terminology and for each collection. So the collection, here you have the terminology, and this is a percentage. And we can see that mesh represents the most widely used represented terminology in the annotation. So then we evaluated, because we have a lot of annotation and we wanted to know if the quality was good. So for that, we randomly selected 50 Medline abstracts and manually evaluated all the annotations in the abstracts. So it represented 6,000 annotations that were evaluated not only based on the term that was matched, but also on the semantics. So whether the term that was matched does its meaning in the publication did it correspond to the definition in the terminology? So the global result is that 90%, we estimated that 90% of, based on the subset, 90% of the annotations were correct and semantically correct. So then we looked more into detail into these results, and we observed that, in fact, there was a correlation between the length of the matched term, so the number of characters of the matched term, and the precision, the probability that it was a correctly mapped. So in other words, the longer the term was, the more likely that it was correct. So one approach to improve the precision of our annotation would be like, for instance, to set a minimal length. So a term, small terms will not be annotated. But of course, if we do that, we lose potential correct terms, correct mappings. So it's, as always, we have to balance the precision and the recall. So then we decided to look at the level, at the results at the level of the terminology. And we observed that so here you have in blue the true positive, so the correct annotation, and in red the false positive. And we could observe that for some terminologies, that for instance mesh, we have very good results. Here it is, very good precision. But for some terminologies, we had many false positives. There were three kind of reasons for that. First reason, for the cell lines and the relation ontology, is that the term were not specific. So I will come later on this. And for FLOPO and ENVO and plant ontology, the main reason was the semantics, the context, because the evaluation was done on man-like abstracts, so biomedical context, and it's plant ontology and environmental ontology, so chances were high that it was not the good context, so the good meaning, the good semantic. So on these terminologies with a high rate of false positive, we tried to see if the approach of setting minimal lengths threshold would be helpful for improving the precision of the annotations. And we observed that for ENVO, it could be a good approach, because many of the short terms, in fact, most of the short terms are false positive, and the correct terms are whole terms with more than 6 characters. This approach could be acceptable for plant ontology, setting a threshold here. So yes, in red it's false positive, and in the true positive, and here you have the length of the term matched. So here, by setting the threshold here, we could still dismiss some false positive, but this approach would really not be helpful for Celosaurus, because all the cell lines are very short terms. So we, of course, here it's a lexical approach, refinement. We also have exclusion lists that are where terms are added case by case. Problematic terms are added to this exclusion list on a case by case approach. I think, also have the idea of adding the prefix, so the database before the accession number, to avoid many mapping that are wrong, because accession numbers are by definition not very specific, like a list of numbers. We also explore other approaches like frequency-based approaches to discard very frequent terms, or syntactic approach to determine, for instance, if the map term is a noun or a verb. And finally, semantic, of course, semantic approach that could be very general, like classifying the journal to see if the context is could apply for given terminologies, and of course LLM embeddings to do like semantic distance rather than lexical one. But we have to remember that this approach, the chosen approach, must be scalable for millions of documents and billions of potential annotations. And finally, we also are trying to extend the annotation to new terminologies, and also creating a pipeline to integrate external annotations provided by partners, and for this also adding a score for the annotation confidence. And we have a current project at Clinical to annotate the clinical case reports with clinical terminologies. So I am going to wrap up this talk with an example where you can search for the interaction of the pteropodidae, and you can easily find that it's infected by a virus that can infect horses or humans, or that is a pollinator of a plant. So yeah. So thank you very much for your attention, and thank you for the contributor of the project. So.
I'm going to make a quick comment. You, Matthew, and I should all talk about using the bioregistry to improve the prefixes for these accession number checks, because they're also interested in doing something like this for Europe PMC.
Yes. In fact, we have interactions with Europe PMC.
Great. All right, thank you. Next talk is from, we don't have time for another question. Stand schedule, sorry. All right, the next is from Melanie Vollmar.
Let's see what I can do with that. But thank you to the organizers for inviting me. This is my first Biocuration conference, and I've sort of stumbled into that field, sort of by accident, perhaps. So my background is actually biochemistry and structural biology. And about 10 years ago, I decided that I want to do programming and machine learning. And then 3 years ago, having known Samir for a while, he said, shall we try and find something where we can combine machine learning and AI to put more information into protein structures in PDBE? So yeah, so I'm an ARISE fellow now, and this is so a little bit of an overview of what I've been doing for the last 3 years. Right, so as I said, I'll give you a brief introduction about the PDB and what we do as deposition or curation, which is slightly different from what we've heard so far. And then why I do the project, what we've developed so far, and where we want to go. So PDB is actually a consortium. It has 3 different members. So you've got a group here in the US spread over 2 sites. There's a group in Japan, and then it's us in Europe. And we are all under the umbrella of the WWPDB. And within the European side, we also have other data resources. So we're not just taking structures in and organizing them and curating them. We also hold the AlphaFold DB, and we have a knowledge base where we put all of the additional knowledge in and information to enrich the structures. And how that looks like is something like that. So we have the intake of computed models, whether they are experimentally determined or whether they are predicted, doesn't matter at that point. And then we lined up with some other tools and other databases, and we sort of linked it all together in a knowledge graph in the PDB AKB, where all of the contributing resources are part of this 3D beacons network. So it's all federated and unified. So whatever query you put into PDB knowledge base will be sent off to one of our data providers, and you can sort of retrieve the data through our portal, as it were. And of course, in order to support researchers, we provide training and outreach so that you can get the most out of the resources that we provide you. So it's no longer just about taking structures in and making sure they are in a unified format, but it's also what additional knowledge can we provide you with so that your structures are far more fun to look at. In the European team, so these are the people that look after the infrastructure and do the biocuration. Whereas the biocuration for us is actually not sort of enriching with Go terms or, yeah, I don't know, finding domain boundaries. That's what we take in from external providers, from other data resources. For us, it is making sure that the data that is behind the sort of the modeling, so the model that goes into the PDB, the raw data that's behind it has actually been analyzed according to community standards, and that the models conform to the standards that we want them to be like. So this is more our curation approach is more towards validation than actually curation. Right, so when we have structures, as I said, we take in some information, but the data resources we take this information in from, they're not necessarily what we want with a structure as a 3D model. So yeah, it's nice to have domain boundaries. It's nice to have some information about an active site, but there's far more that actually a structural biologist is interested in. And what happens is when they look at their structure and they describe it, you have these beautiful photos and pictures in the publication where they look at their model from all kinds of direction, and they describe the intricate networks of the residues that are involved in whatever that is in that picture, whether it's an interaction site with another protein or a ligand or whatever. So they really describe in detail in the text what they see. But because it's in the text, it's unstructured and it's no longer accessible, and we can't do anything with it. This is the common narrative. So everything is beautifully described, but it's just dumped there and nobody can do anything with it. So we much rather would like to have it in one of the databases. But as I said, the current curation, using manual biocuration, you all know that is not scalable to the amount of papers that come out and to the amount of structures. Yeah, it's also kind of like depending on sort of what resource it is, they have their own specification, and it does not necessarily cover what we want to do with the structures. Nevertheless, I looked at, what people, for example, in UniProt would do. And so, yeah, followed their kind of like reading a paper, extracting the knowledge, writing it in a database. And I thought, so how can we now do that with an algorithm? Can we sort of mimic some of that to at least get some information out that would be useful? Because the team of biocurators we have, they were so busy with figuring out that the structures are correct, that they don't have any capacity to read papers on top of that. We also can't offload it to UniProt or any other resource because they were busy too. So we try to mimic this sort of process, let a machine algorithm read the paper, extract the sentences and write it into a database. Yeah, and human in the loop, as we've also heard the entire, throughout the last few days, is very important to make sure that it doesn't write any nonsense into our database and we don't have a propagation down the road. So the general workflow that we employed was actually when people deposit a structure, they usually do that in connection with a publication. So we know for structures that are mentioned in the literature for the first time, we have around 80,000 publications available. But not all of them are necessarily curatable. So some of them may just mention a structure in passing because they've just happened to fall out of some other process. And others write really intricate descriptions of what's going on. So I used LitSuggest as a filtering tool, and then also decided we actually don't just want to have the abstract and the title, but the full text. So we ended up with about 20,000 publications that should be curatable and get us information out. 10 of those were selected at random, and we did the manual annotation of the terms that we thought were relevant to describe a protein structure. And that was done again in an open tool, in that case TeamTat. And we just basically had to provide the PMC IDs. The tool would load the publications and we could just go off and annotate. So we devised an annotation guide, how to use the software with examples. And then for 4 months, the Biocuration team and myself sat down once a week for 2 hours remotely, because we were in different time zones, and annotated the 10 publications. In a blind fashion. So we had pairs, but nobody knew what the other one was annotating. And once we closed the annotation round, so we agreed with we were just under 80%, we had full annotative agreement, which is the thing is very good. So our rules worked quite well. We had a similar background. We knew what we expected. And then we did some tidying up and trained our initial model. So because we are obviously limited with resources, I had to do that step by step. So once I had my first model, I then picked another 10 publication, let it annotate, cleaned up the annotations based on our guidelines, and did it iteratively to actually annotate 40 papers in the end, and get actually a best model that does that very nicely for me. Additionally, I had another 10, which were completely unconnected to whatever we did before, to have a benchmarking data set. Also, because with this iterative process, the test set and the training data, they would change every single time, because I always increased the amount of data that was available. Right, so here's sort of the performance that we got with the training, and you can see increasing the amount of data reduces overfitting and gets us the algorithm more stable. Depending on which entity type, we had 20 in the end. We chose, we had a variety of position and recall scores, so that's across all of the entity types, and the ranges are in the brackets. And when I use the benchmarking data set, I get these scores from my best model. So I think it does reasonably well. So if I relax it a bit and I get a partial score, then sort of around 90% of my entities are correctly identified. The model was deployed as a small publishing company. So what we do there is it runs in the background and annotates the publications. If it finds a residue, it highlights that, and there's another pipeline wrapped around it that allows us to then link back to the PDB structures, and actually also the validation statistics that give you information on how good the structure is and how good that residue is interpreted and supported by the data. So you can look at that in line while you're reading the paper. So you can look at the structure while reading the paper. So far we have had an intake of these annotations provided by the publisher. We are about to finish our own in-house pipeline to roll it out across all the publications that somewhere, somehow mention a structure. As long as there are open access, that's our prerequisite. Write it all in our database, and then you have this beautiful table. And also in preparation is a RESTful API, so that way, if you wanted to, you could actually just pull that data yourself, and then you could do something with it. And this is all open access. Right, so what do we want to do in future? Besides just having the API, we would like to integrate that in some form of a sequence viewer, which is also underway because we're redesigning the web pages, and also combining it with our existing resources under the frameworks of BioCypher and BioChatter, which are both completely open source resources, Python-based, where you can create knowledge graphs and plug them into a system that allows you to plug and play with LLMs to interpret the content of your knowledge graph. And with that, we hope that people can do some hypothesis generation, but also if we expand further, not just on structure papers, but any paper that mentions the residue and an experiment on the residue, we could use that to actually validate predicted structures Because currently they are only looking at whether the model has done a good job, but nobody actually knows what it predicted, what it created is actually what is biologically relevant, what has any sort of, whether it's how it will be in an organism. So we try and do that by probing it with non-structural results. Yeah, and that's the acknowledgement, that's the team, and it's my last slide.
Thank you. We have time for 1 or 2 questions.
I appreciate it's different.
Thank you. So how can publishers help to this approach, right? So what can we do?
You can come to us, talk to us. The models are freely available. We have the code for pipelines. You can see what needs to be done to work within your own system. But yeah, this is all open source, so you can come to us and we can do that. And we have systems in place now how you can deposit back your annotations that you find. So because we already had to do that for our collaborator. So yeah, happy to take people on. Oh, it's the last question at the top.
This is great work. So one thing I was trying to understand is, so you're a PMC. So technically what you're doing with the publishers, you technically could be working, or maybe you are working with Europe PMC. So I'm from Glygen, and we have had some discussions with them. And the idea is that can we connect directly using some of the methodology or similar type of methodologies, using dictionaries, for example, to PMC, then which connects to Glygen and other resources to make it more rich, what people get from PMC and other places. So any thoughts on that?
Yeah, so Santosh here, who is an acknowledgement, so he's at Europe PMC and he was involved or sort of gave us some help with the model training. They know where the model is, they were involved in it, they can take it in, and that's what they're planning to do, to add it into their own pipelines, because they have slightly, different access to publications as we do, because we can only use what is open, but they have more access. Generally, they also have a way of submitting annotations, so they have a standard process and a standard form, where if you have annotations, you can submit them there and they will happily take them in, or you just, contact them and talk to them and they will be happily talking, and discussing that.
Thank you again. Last talk of this session is from Sybilla Corbett.
Hello, my name is Sybilla Corbett. Oh, is it? They're not quite. Let's figure this out. Maybe if I take this off. Pop it up here. Better? Fabulous. My name is Sybilla Corbett. I'm going to be talking about ChEMBL, which is the database I work on as a biological curator. I'd like to thank the organising committee for accepting my talk. I first attended Biocuration in 2017, but this is the first time I've spoken at one. A bit anxious. So I'm going to start with an overview of ChEMBL. I wasn't really sure what the kind of base level of knowledge was, so I thought I'd start at the beginning. We're going to talk about how we structure our data and how this kind of aids the deposition of diverse data. And then I'm going to kind of segue into AI readiness and how we've been using we've kind of done a small project using natural language processing. I did present a poster about the natural language processing yesterday, so I'm not going to go into a great deal of detail today, but anyone who's interested, please come and find me. I have the poster, so we can have a look in the chat and talk about that. So what is ChEMBL? It's a chemical biology database from EMBL EBI. There are about 40 open source databases at EBI, and these are the 4 that are managed by the chemical biology resources, which I work for. So we've got ChEMBL, a database of small molecules relevant to biology. We've got ChEMBL, a database of kind of quantitative small molecule bioactivity data. Unichem is a useful way to link resources using chemical structures, and then ChEMBL contains patent data. All our resources are open, and we try to follow fair data principles. So ChEMBL celebrated its 15th birthday last year, and it was handed over from a private company to ChEMBL, who kind of developed it and now maintain it. So we're utilized across academia and industry. We capture information from all the stages of the drug discovery process. So the first kind of 4 here describe before even you get to kind of clinical candidates, but it describes the structure activity relationships of these molecules. Oh, that's slightly wrong. We actually have 2.5 million compounds now. So the data in ChEMBL is mainly manually extracted and then curated, and it's kind of structure activity relationships, as I said, from medicinal chemistry and pharmacological data. Literature, sorry. We do take deposited data sets and from a wide range of different institutions and consortia. Data deposition is a really growing area for us, and we would encourage anyone with relevant data to please submit it to ChEMBL. Our data is released intermittently, usually 2 or 3 times a year. We're currently on version 35, and we should be releasing 36 in the next couple of months. So the ChEMBL data structure. These are our key entities, and they are stable identifiers that can be used to search either the interface or to programmatically query the database. What's happened over the last 15 years? Well, ChEMBL has grown in size and complexity and the diversity of the data that we store. So I've got here, this is the database schema for ChEMBL version 1, and this is the current externally facing schema for 35. We have even more tables that we keep to ourselves. So there are 79 user exposed tables in kind of 8 notional subject areas. So any changes to this schema are announced in the release previously, so that people have time to kind of update their processes and know what's coming up. So as I said, we have a kind of semi-manual curation. There's 2 overlapping routes for data. So there's the compound, the chemical curation, and then there's the biology curation. We have 3 curators, and our job is to make sure, as we are all aware, the data is as accurate and coherent as possible. So all of these processes are captured in Jupyter notebooks, which kind of talk about different target types. And we think this makes it very transparent. It's very easy to pass from curator to curator, and it also makes sure that the way we're curating things remains stable from one release to the next. So this is our sort of how we get drug data in. So the United States adopted names and the INNs and the clinical trials database are used to identify compounds that are kind of in development, and then we get information from the FDA and the EMA on approved drugs. And we extract mechanisms and indications, we calculate properties, and all of this is kind of put together in the drug cards that are on ChEMBL. And cross-references are included for all of this information. And this feeds into ChEMBL, and then also feeds into open targets. So everything I've spoken about so far allows us to accept the deposition of diverse data sets and makes us a unique resource for questions about medicinal chemistry. And I have a few slides to talk about what I mean when I'm talking about the diversity of our data. Oh gosh, I'm going very fast, aren't I? I'll try and go a bit slower, although everyone will probably like a coffee. So ChEMBL version 9 was released in 2011, and it had 6 data sources. And this this completely unreadable graphic over here is the 48 data sources that we currently have in ChEMBL. So there's been quite a lot of data deposited since then. You can see there's a sort of quite a steady rise in literature data and some quite big jumps in our deposited data. The highest number of assays and compounds that we have comes from the kind of the medicinal chemistry, the pharmacology literature. But the most activity points we have actually comes from our deposited data. And that's not surprising because we've got some very large consortia that we work with, and they tend to take a small number of molecules and really, really heavily identify what's going on and extensively test them. So that's an interesting thing. So I'm going to talk about two examples of recent depositions that I've chosen to highlight because of the kind of complexity of the data and the size of the data, and also just how different they are from some of the other stuff we store. So we're part of the UB Open Consortium. They have a focus on chemical probes, cell active, selective, and highly validated research tools. So they've taken them and characterized them in a number of different ways. Our role in the consortium was to store the data in a database and also to create an interface for them, a kind of the UB Open gateway where the data is displayed. So all of this data that we get from the consortium is fed into the ChEMBL database, that goes into the Elastic Search Index, and then the search index puts it both into the ChEMBL web interface and also into the gateway. So it's not a kind of duplication of effort. Oh yeah, one thing I wanted to say, over the last 5 years, these guys have deposited 36% of our deposited data. They're a huge contributor to ChEMBL now. So this is a kind of something completely different. This is a kind of slide about some data that we have on drug metabolism data. So the group of Michael Zimmerman at EMBL Heidelberg work on drug metabolism by the gut microbiome. It's really interesting data, giving insights into how gut microbiome kind of affects human health. And the understanding of how different bacterial species can metabolize drugs is interesting both to advise medical therapy, but also drug development. So a little bit about the actual point of the session, AI readiness. So obviously, we've heard it over and over again, garbage in, garbage out. High quality data is fundamental to these artificial intelligence applications. And this has been specifically recognized for drug design. So I've spoken about how we structure our data, but there are more ways that we kind of make it usable. So we're very aware that fair means computers as well. And it's our intention, I mean, these are kind of some of the checkboxes that we have, you know, we're all aware of how you make data fair, I guess. But we're planning on having a series of kind of user group meetings to try and regularly talk to our users from all their different backgrounds and understand the ways in which we can help them use our data. One of the things I'll talk about, every ChEMBL everywhere all at once. Before he joined our group earlier this year, our new group leader, Noel O'Boyle, had been a very long time user of ChEMBL. And to celebrate our 15th anniversary, he downloaded the first 34 iterations of ChEMBL. And he was able to extract from them smiles and inchies and identifiers. And he wrote a blog post about it. So the first place people mostly land is on our website, but all of our data is available through an RDF platform or programmatically via the API endpoint, or can be downloaded in its entirety. We have guidance on all of these data access routes in our documentation. So we try to help wherever we can, and we also have a very active help desk that often fields queries about these different access routes. So we hope we're moving in the right direction. Our data is already kind of quite widely used for AI and machine learning applications, especially in the field of quantitative structure activity relationships. But that doesn't mean that we can't improve, and we have lots of plans in place for kind of utilization of ChEMBL. So I'm going to talk about a very small project that we took on. It was a side project for all the people involved. So, but we wanted to use named entity recognition to pull data out of our assay descriptions. Our assay descriptions are a free text field, and we thought that by using NLP, using existing tools like Spacy and text-to-term, we could pull data out, map it to ontologies, and make it more machine findable. And this was the subject of my poster, and I'd be very happy to talk about it with anyone who wants to, because I'm coming to the end of my slides. So yeah, I think the database the size of ChEMBL is always a massive group effort. And I'd like to acknowledge our funders and all the wonderful people in my group. I'm in my 8th year at ChEMBL. Oh no, at EBI, not at ChEMBL, but there's such a lovely group of people to work with. It's such a lovely place to work. If anyone sees a job advertised, do apply. And that is all I wanted to say. Thank you very much.
Thank you. We've got plenty of time for questions.
So welcome to the ISB. You did great, so no need to be with this. Long time ago, when I was referencing various chemicals in the previous life, I had to choose whether to reference the ChEMBL entry or the ChEMBL entry. Now that was over 10 years ago. Maybe the answer has changed, but could you give some advice as to how one, as either a user or a curator rather, might choose one versus the other to reference? And same question as a place to deposit information about a chemical.
Okay, it's a good question. I think it depends on your use case really, because I mean, ChEMBL is an ontology. So if you're kind of looking to reference an ontology to kind of bring structure into the referencing of your chemicals, then I think ChEMBL is probably your answer. But ChEMBL's a repository for data. So if you've got a chemical, a target, and some bioactivity data about it, then that would be best brought to ChEMBL.
So given infinite text mining curation powers, what would ChEMBL like to capture as structured information that isn't already capturing, either about chemicals, assays, publications, so on?
Oh, this is very interesting. So yeah, I mean, the project that we've done is very backwards facing. We're looking at our old data, our legacy data, and trying to pull more information out of it, especially because we're very aware that the earlier assay descriptions are nowhere near as descriptive as our current assay descriptions. I mean, with infinite possibilities, at the moment, our data extraction is done by an external company called Molecular Connections, and they pull activities and assay data and structures out of papers we have 7 core kind of medicinal chemistry journals that we pay them to give us information from, and it arrives twice a month. I mean, if we, I'm sorry, Molecular Connections, if there was a way we could cut them out of the loop, it would save a lot of money in the long term. I mean, we're talking about job losses. But yeah, I think, I mean, given infinite possibilities, to have something pull that data for us would be fantastic. And I mean, at ChEMBL, what we do now is kind of what we've been talking about in the AI sessions. We are now validating more than, we're not doing the extraction, we're validating information, we're putting sanity checks on it, we're taking, biological knowledge and saying, does this make sense? So yeah, that's my side. I hope they're not listening. They do a really, really good job.
Hi, I'm a longtime user, particularly of ChEMBL, but to a lesser extent ChEMBL. But because this is such a strong database with lots and lots of information, I am struggling with my use case is toxicology or environmental chemicals, but in the same types of assays that medicinal chemists or other groups would use. So is there a space for this kind of information, or can we expand either ChEMBL or ChEMBL to include environmental health kind of chemical exposures?
So I mean, if we're talking about the same sort of experiments that are being done on all these other chemicals, I don't see why it would be a problem. I mean, we've not got it yet, but there was a pesticide project in Michael Zimmerman's lab that we wanted to bring in, and they just haven't got around to structuring the data for us yet. I mean, you've seen our data structure. It's huge. Has anyone deposited to ChEMBL? Okay, it's really lovely and it never goes wrong. But yeah, there's, I mean, we like data. If the reason why we might not have it is because it's not in the journals that we've been looking at, and the people who are making it haven't kind of decided to deposit it with us. But I mean, if you can give us a chemical and a target and an assay, we're usually pretty happy to try and fit it into our data structure.
One more quick question is, what is ChEMBL's approach to monitoring the drug development and drug approval there, for instance, clinicaltrials.gov? Like, how do you keep track of the updates?
So there is a database within the database. It's a hidden database. It's called DrugBase. And there is a team that is devoted to DrugBase. And DrugBase releases once a year instead of twice a year because it's such a huge job. And they scrape data from clinical trials and they download the FDA Orange Book and they get information from the EMA. So yeah, we're kind of pulling in all that information from the websites in whatever way we can get it. And some of them have APIs, but most of them don't have APIs. And then that is all kind of processed as drug data or clinical candidate data. And yeah, bless you, sorry. And then that, yeah, so we have a pipeline for it. Did that answer your question? Yes.
Yeah, it's partly automated.
Yeah, there's workbooks that they run through, but there's always sanity checks, and I mean, there's always exceptions to every workbook that you run. So there are people who do that full time.
All right, thank you, Sybilla, and thank you everybody who talked from the Natural Language Processing.